Heart disease research is often performed in small animal models such as rodents. However, the physiology of the human heart differs significantly from that of the rodent heart.
Our protocol uses cardiomyocytes differentiated from human iPS cells to model ischemic heart disease and to quantify the damage and functional impairment of ischemic cardiomyocytes.
This protocol may provide a convenient method for personalized drug screening using iPS cells derived from individual patients.
The appropriate handling of the iPS cells, such as in using fresh medium and changing the medium slowly and punctually, is critical to ensuring a successful differentiation into functional cardiomyocytes.
Demonstrating the procedure with Yunliu, Liang, and Mengxue will be Chen, a PhD student from my laboratory.
To induce human-induced pluripotent stem cell differentiation into cardiac cells, first coat the wells of a 96-well culture plate with 100 microliters of 1.675 micrograms per milliliter of laminin in PBS. After a 30-minute incubation at 37 degrees Celsius, seed 3 × 10⁴ stem cells in 200 microliters of iPS maintenance medium, supplemented with 10 micromolar Y27632 into each well, and return the plate to the cell culture incubator. After 24 hours, replace the supernatants with 200 microliters of iPS growth medium per well, and return the plate to the cell culture incubator for an additional 2 to 3 days. When the cells reach 70 to 80% confluency, replace the medium in each well with 200 microliters of pre-warmed differentiation medium A, and return the plate.